Hello, everyone. Thanks for coming. I'm Amy Elliott. I run product for FormBio, and I'm delighted to talk to you about some of our recent advances with our AI technology for gene editing, microRNAs, and gene replacement. So at FormBio, we think of our mission as transforming the future of genetic medicines and genome engineering with AI. So what our history is, we started with AI-enabled genome engineering at Colossal Biosciences. So FormBio was really incubated within Colossal as the computational biology arm. So our expertise is very much a mix of everything from molecular biology and software engineering through to bioinformatics and computational analysis. So what that gives us is a very broad set of expertise for building foundational models for genome engineering. And so what we decided when we were still within Colossal was this is actually a quite generalizable set of technologies. And so we spun FormBio out in 2022 so that we could focus on human health and bring these technologies to the place where we really want to see them shine for patients. So on the right, you can see sort of where we are with providing our optimization and prediction and computational analysis tools with respect to cell and gene therapy. We've done over 60 gene replacement constructs. We've done more than 20 gene editing constructs, gene silencing, and we have recently started moving into CAR T-cell optimization as well. And I'll talk to you a little bit about how we approach those different problems and what that means directly. So the way that we think about successful gene therapies from the perspective of the data and the computational side, there are three sort of important pillars for success. It has to be safe, right? We want to reduce the immunogenicity. We need to have the lowest possible dose, lowest possible toxicity, minimal contaminants. Efficacy has to be as good as possible. That means the correct genome identity has to be in place. You have to actually deliver the therapeutic genome that you're trying to deliver. Tissue-specific targeting is really important, and that's where the capsids come to bear. Durability of expression is something that we're still learning about as a field, but that's going to be also really important, especially as we're treating younger patients. And lastly, manufacturability, and this is where our technology has been really shining. So getting a sufficient titer during manufacturing, making sure that you have reproducible genome integrity so that you're, again, delivering the correct genome, and scalability and lot-to-lot reproducibility. That requires a lot of coordination with both upstream and downstream processes. And so as we're thinking about how we optimize the process for purification of these therapies and enrichment and things like that, what we're trying to do at FormBio is shift that entire conversation left and upstream. Let's think about how we design these constructs so that we have fewer problems at manufacturing, so that we don't get to the clinic and find out that, gosh, we have a lot of not empty, but full of not the right therapeutic capsids. So FormBio, because we're a computational place, we see ourselves in a pivotal moment with this industry for the combination of data and analysis and AI. So multi-omics data, whether we're talking about whole genome sequencing or we're talking about long-read sequencing to characterize the AAV drug products, or we're talking about RNA-seq data to look at capsid profiles, the availability of big data sets really unlocks the capability of AI. Machine learning and deep learning models need a lot of data to be able to do something that's useful. And so these technologies at the confluence of the ability to do compute for AI models has really been a critical unlock. But those are also only useful if the data is good and if you understand how to interpret and use the data. That requires a quite multidisciplinary team of expertise. So let me give you an idea of what that means from the way we look at this data. So the graph on your left, on the left, is a graph showing, this is a representative subset of the data that we have. So on the y-axis, you're looking at the number of sequencing reads. This is just a subset of PacBio long-read sequencing data for gene replacement vector designs. And then over time, how we've collected that data. So we have hundreds of millions of reads worth of sequencing information across a variety of different vector designs. The things that we are learning looking at data and different construct designs at that scale are incredibly useful lessons that generalize quite well. So for example, the genomic context matters. So if you take your coding sequence and you put it into a vector that has a variety of different regulatory elements because you need to achieve different biological goals, what you'll find is that if you use the long-read sequencing technology to characterize that product, you will see a quite broad response in terms of your full genomes and partial genomes. So the figure on the right is looking at that relationship between full genomes and partial genomes. So a full genome in this case, right, is everything from one ITR to the other, regardless of if it's a self-comp or single-stranded, has to be intact for it to be as therapeutically efficacious as possible. And what we want to see is that we have a lot of full genomes and basically no partial genomes. So that'll be the green part of the graph. What we see in practice is a really broad distribution of real data for different construct designs. And it matters where you, where you are in the process, because as you get the process figured out and the protocol streamlined and everything else, you start to see more reproducibility in terms of where you fall on this graph. But doing the, moving up the graph to the green part requires doing the hard work at design. So the way that we analyze this data, and this is another thing that's come out of a lot of our expertise looking at a lot of different species worth of data at Colossal, is being able to interpret the results from the long-read sequencing is not just an exercise in a GitHub pipeline, right, that's available for free for bioinformatics. It's also being able to understand the context in which those results are meaningful. So we're doing things like looking at the residual plasmid DNA and the host contaminant. We're looking at how much RepCap and helper got recombined into the drug that was actually made, right? We're looking at therapeutic genomes to make sure that the mutation space is small enough that you're still actually getting the protein that you intended to deliver. These are really important characterization pieces to make sure that you understand the drug that you're delivering. The high resolution that we get with sequencing is a really key piece of allowing us to do molecular design and optimization. And that's where our AI technology comes in. So what we're doing at the end of the day is we are doing two things. We're taking all of that data that we have built a foundational model on, and we're predicting where we're going to have problems in the sequence for manufacturability. So we can look at swapping out different promoters, looking at WPREs, the different regulatory elements, and we can see what kind of profile we're likely to get in manufacturing. That's incredibly powerful for determining in silico a screen for thousands and thousands of different construct designs. And then for the coding sequence, the way that we're doing codon substitution is the key piece for optimizing these genomes. And the reason for that is because there are trade-offs, right? You can't have both a maximum codon adaptation index and minimal changes from the wild type and reducing all the truncations at the same time. There are only so many codon swaps that you can do, which is hundreds of millions of them. So what our technology does, the key thing is it figures out where we need to make the codon swaps and what we're going to swap it with. And that's the key piece for AI/ML. So I wanted to give you just a little bit of a flavor of the data that we use to not just validate our technology, but also the real-world outcomes that we're seeing in the laboratory. So this is a graph that's showing that as our model has evolved, as we have gotten better at understanding the data, and as we have built up our training corpus to those hundreds of millions of reads, you can see that on the far right, the curve is shifting to the right, which is how we measure our accuracy and our confidence in the model. Lastly, and then I'll get out of computational land, I promise. What I want to show you here is the correlation for our predictions between the predictions on the left and the truth on the bottom. So everybody does, hold out, right, data sets. So you're going to train your model, and then you're going to test against an entirely new data set. And so what we're seeing here is about a 0.83 R squared for our predictions versus our truth, which is really good for AI. If it was 93, I would be concerned about massively overfitting. So we're living in a space where we are confident that our predictions are going to be not 100% spot on, but they're going to be very good out of sample. So the way that we deploy this technology for genome engineering is in a couple of different places. At the rational design side, that's where the predictions come in. We're going to evaluate the entire construct together to see where we have problems that need to be changed. Sometimes that's going to be a promoter. There are several promoters that are known to be problematic for truncating during replication. And so we end up with a lot of partial genomes, which are, if not toxic, then non-therapeutic. Similarly, the context in the entire construct matters. And so what we can do here is we can say, all right, well, this is what it's going to look like. Let's try 1,000 different other options with different regulatory elements that are still going to have the right tissue specificity, et cetera, et cetera. And so our technology is very fast for designing these different things and then evaluating them. And then for the codon optimization, again, what we're doing is using our deep learning model to select where and what change to make. And then for capsid selection, we can also evaluate these ITRs to make sure that the ITRs that we're using are going to be as good as possible for packaging. And that's the extent of where we currently, where the model currently sees. But we also do RNA-seq analysis for selecting new capsids, and that gets us the feedback loop with the whole genome. Okay, so let's talk about outcomes. So one of the things that we're seeing very, very routinely is we're able to increase full genomes. That out of sample 0.83 correlation means that even if we have a gene of interest that we have never seen in our training corpus, we're still doing a very good job of improving the full genomes. FormBio is in the black bars, and our customer client is going to be in the blue bars. We're always comparing against a control. For efficacy, here we're looking at the copy number. We're seeing about a 5x expression increase most of the time. Importantly, you don't always want to overexpress. Sometimes that's toxic. Our model also has the capability to tune down expression. So really what we do is we work with clients to determine what the target profile is for what we need this genome to do. And then we will set the model objectives to make sure that we achieve whatever those goals are in the best case scenario, given that there are going to be trade-offs between the objectives. So lastly, increasing titer. Titer is a tricky one. But we're still seeing almost a 2x increase in titer most of the time. Let me give you a specific case study example. So this was a terminated program that we worked with a client on. They were getting about 15% full genomes measured by long-read sequencing, which was just not sufficient to go forward. That's too risky, right? And so we worked with them to screen more than 200 different constructs with a mix of promoters and other regulatory elements. So we selected the top 3 of those, and then we optimized that coding sequence. They were concerned about making sure that there were minimal changes from the wild type, which put an additional constraint on what the model was able to, or allowed really, to optimize for. And so what you can see is that once we get through with all of that, we end up with a 3 to 5x improvement in genome integrity. We ended up in this case with a 2.4x increase in titer over the previously non-viable program. So what we've done here is we have taken a construct that was absolutely not viable to take forward into manufacturing, and we have made that, we have provided a path forward. And this is the kind of thing that's really exciting for us. MicroRNAs are something else that we're getting into right now. So our model knows things not just about the sequencing data from the reads themselves, but also physicochemistry. And so we are able to do things like evaluate these structures for complexity, and we can optimize to reduce the complexity enough to permit things like synthesis and packaging without running into replication errors or synthesis errors. And the last thing that I really want to say I'm very, very excited about is we announced a partnership with Cure Rare Disease to advance a Duchenne muscular dystrophy program, where we're working together on a gene editing construct that we hope will be our very first sort of joint clinical product. And so this is moving right along into in vivo studies right now, and we're really excited to see where it goes. So stay tuned for that as we develop that construct. And what I want to leave you with is one thing that's been very inspiring to me about working in the cell and gene therapy space is the community. This is a community that is far more collaborative and far more interested in working together to advance the entire field than a lot of other places I've seen. And so we work with an AAV working group, which is a consortium that we've pulled together with, you can see all the companies in the box and a variety of other academics. We meet monthly. What we did last year together is we published a set of standards for how to do the bioinformatics pipeline. What we're working on now is the library preparation. It is the wild, wild west for long-read sequencing technologies in terms of the different chemistries and how we approach the problem of using those assays to understand our drug products. And so this group is working together to really make that standardized and accessible to everyone. Thanks for your attention.
Thank you, Form Bio.